Then what we're going to do now is talk about cell survival curves. Um, this is a somewhat complicated story, but I don't think it's that complicated. As this progresses, you will see we will use cell survival curves for everything. And so understanding this topic is going to be important, but there will be some things you'll learn here that will become emphasized in a different way later and will probably help in understanding. First, the easy stuff. We already said most of this. The cell has a nucleus in it. The nucleus is where the DNA resides. And while there's DNA out here in the mitochondrion, the DNA we worry about most and that we try to hit most in order to cause cells to die, tumor cells to die, is nuclear DNA. The nucleus is important because that's where most of the genome is. And any genome that's elsewhere is probably not nearly so sensitive because there are many copies of it one way or another. Okay, so I'm going to bring up a concept now that I hope makes sense. If not, please bug me. In the biochemistry world, when people want to measure cell death, they often use a dye method. They add a vital dye. If the dye gets in, the cell is dead. If the dye is excluded, it is the cell is dead. Trypan blue is the most common. There's another one called nigrosin. And what it does is these measure loss of membrane integrity, but they sort of measure ability of the cell to metabolize. If the membrane is not intact, the cell's not metabolizing. But a second way of being able to measure cell death is more tedious, but it relates to cell death in a different way. And that's using reproductive cell death as a measure, which some people call clonogenic survival, clonogenic assay, cell survival curves, capacity to reproduce, or in the opposite, the loss of reproductive integrity. Now, what are we measuring when we measure reproductive cell death? We are measuring whether the cell has the ability to divide and in culture form colonies in the patient, make the tumor grow. So why is this distinction so important for us between is the cell metabolically dead, as we were measuring a trypan blue dye assay, versus is the cell reproductively dead, as we were measuring a clonogenic survival assay? It's important because when we think about killing tumor cells, we actually don't care if the cell sits there and metabolizes all it wants. It can't ever eat up enough nutrients all by itself to do anything unless it is dividing. So a metabolically active cell that cannot divide, a cancer cell, is not a cancer cell anymore because it can't divide. In the end, all that we care about is whether we've made the cells reproductively dead. It takes a higher dose of radiation to make them be metabolically dead than it does to make them be reproductively dead. So now think about what we do in radiation therapy. We treat patients, we're trying to kill the tumor. We're actually trying to make the tumor stop dividing, make it not be able to grow anymore. And if it sits there and metabolizes, who cares? Yet for us, getting the best dose to make the cancer cells not able to grow, but not damage surrounding normal tissue, is actually our greatest goal. You will be able to kill any tumor that walks through your door. And you know what? You'll kill the patient too. The hardest challenge of radiation oncology is to give the dose that kills the tumor and spares as much surrounding normal tissue so the patient still can live life and is okay. So for that reason, radiation oncologists and radiation biologists distinguish between trypan blue dye exclusion, metabolic death, and reproductive cell death or clonogenic survival. So therefore, when we are measuring whether a cell can survive or not, we are measuring whether it can divide, not whether it can metabolize it. This is a much more tedious assay. It's a much trickier assay, but it is the one that is relevant for radiation oncology. I would argue it should be relevant for chemo too, and chemo people should be using this. Some have gotten the message, but some still don't. Okay, now, and this just says everything I just said. We will talk about the different kinds of cell death that would be important here, and that will become significant. Okay, there is a limitation to this, and that is it works great for tumor cells. It works great if we're looking at normal tissue toxicity, provided the cells are capable of division. If they're not capable of division, if they're highly differentiated, we're not going to be able to use cell survival to monitor it because no cells divide to be able to see it. So a lot of times what we will do, and we'll see this later in the course, is we might look at like loss of function. So for instance, with lung cells don't divide in culture. So if you want to know whether you're inducing an effect, you might want to measure breathing rates because that relates to how much damage you're getting that results in fibrosis. So it only is going to relate for dividing cells, not for differentiated cells. What the idea was originally was, whoa, let's just take every tumor out of every patient, check them for how they respond to radiation, and there we go. We know exactly how we're going to treat. What was the problem with this idea? Tumors do not grow out of every single patient. They tried to do explants. They didn't work very effectively. So people kind of gave up on it, moved into looking at established cell lines that are different types of tissues, have different patterns, different species. And they said, look, if we know these things, we can probably guess how this tumor is going to respond. We use other methods for determining tumor responses now. We can, in some cases, culture in vitro. It's hard. A lot of times what we do is we take the tumors right out of the patients and plant them into a mouse and look at survival in a mouse. And we'll talk about those methods as well. But for now, we're dealing with cells in culture. And there are multiple different cell types that we use in culture. One is the perfectly normal cell. There are some that do divide. Fibroblasts, lymphocytes can be forced to divide in culture. They have a limited lifespan, so they're only going to be useful for a given period of time. There are cell lines that have been developed, and these are actually extremely helpful. Some are transformed cell lines that are, like a cancer, and some are untransformed, so they behave much more like normal cells. There are then clone cell lines where they have maybe a particular feature, radio resistance, radiation sensitivity. And we can look at those. And then finally, there are rarely used some hybrid cell lines where you fuse two cells together and they may have some genetic components of each of them. Okay, so here we go. This is the method of the cell survival curve. Now, this method is important for you to know. There are going to be some questions on the boards that relate to this. So we're going to go through it in a little bit of detail here. What happens is, in this example, they're growing cells on the bottom of a tissue culture flask. They add trypsin, and actually they would add EDTA usually. It lifts the cells off the bottom of the flask, puts them into suspension. And now what do we want to do? We want to see whether a single cell can become a colony of cells. So we plant these cells in a petri dish, one by one, and we allow them to grow for a few weeks, and then we count the colonies that result. This first example is for the untreated cells. 100 cells are planted down. They're given no dose of radiation, but we incubate them for a couple of weeks. We go back and count the number of cells, number of colonies that we see. So those are the number of cells that we're actually able to divide into colonies. So we're doing that sort of clonogenic survival method. And the answer is here that this is the plating efficiency. How many cells that you plated from your original culture were able to grow into colonies? The answer here is 90%. Planted 100 cells, got 90 colonies, 90%. 90% of the cells that were seeded, they grew into colonies. So this is the plating efficiency. Now we're going to do the experimental part of it. We are going to give different doses of radiation, and we are going to ask how many of those grow into colonies. But the trick here is that as we increase in dose, we know that we're killing more and more cells. So therefore, we have to actually plant more cells per higher doses. So 2 gray, we're going to plant 400 cells. And in this example, we get 72 colonies out. This allows us now to plot the first point in our surviving fraction, which is the number of colonies that were counted, which is 72, divided by the number that were seeded, which is 400, times the plating efficiency, which is 90%, and we get 0.2. We do this for 4 gray, but now we have to plant even more cells. We do it for 6 gray, and now we have to plant even more cells. But in each case, we calculate the surviving fraction, which is 0.2, 0.4, 0.005, and we can now put that into a curve. Now let me show you, this is another way to do it instead of doing it on single plates. Some people do it in multi-well plates. My lab tends to do like multi-well plates a little bit better. And we use this to develop a survival curve. Here are what these colonies look like. Small colony, big colony. Here's what they look like on the plate. And you have to count all these. Again, on the plate. I don't know why that's there. And this is not important. We'll talk about this later. But here is, here are the data on what happens if you plate too many cells. So the fact is you have to know something about your dose. If you thought here that the number of the dose would kill only a few cells, plating 400 cells and having to count 390 colonies would be a real pain. So you got to know what 2 gray is going to do in order to plant the right cells in each approach. So this table is just saying, okay, if you believed that 5 gray killed 50% of your cells and you planted 100,000 cells, you're going to have to count 50,000 colonies at the end. I mean, you know, I don't want to have to count 50,000 colonies on a plate. Even with the most sophisticated plate reader, you're not going to get accurate numbers. So you actually have to know something about your dose before you even start doing these experiments. I like the next slide better. I think it's a little more accurate. So here's now the survival curve. So we take those surviving fractions that we get, and we plot them against radiation dose. Let's just go back and recapture that for a moment. Here we go. These are the surviving fractions now that were generated here, where we took the number of colonies that we counted, 36 here, divided by the number of cells that were seeded, which is 1,000, times the plating efficiency, which is 90%. We get 0.04. Here are the numbers that we get for the surviving fraction. Now we come back to our curves, and we plot surviving fraction against dose. And we get a curve that looks like this. Now, this curve is interesting. It's asymptotic. It doesn't ever really touch the x-axis here. Let's look at it better here. But here's the problem with this curve. The effective dose to kill 50% of the cells is shown here. The effective dose to kill 90% of the cells is shown here. Now think about that clinically. If you start out with a tumor that's 10 to the 9 cells, and you kill 90% of the cells, you're going to be left with 10 to the 8 cells. These doses are not realistically what we would use in radiation oncology. The doses that we would realistically use are way out here, which we can't really see on this curve. So this linear by linear plot then is not considered to be all that accurate or useful. So instead, people converted to what's called a semi-log plot, where it's the log of the surviving fraction against the linear radiation dose. This log linear plot gives us a curve that looks like this, where out here we can be looking at the effective dose to kill 99.999% of the cells, something that's going to be much more important to us clinically than killing 90% of the cells. Now, I want to point out something here that we will talk about, that I will keep pointing out throughout. And that is, if you look at this curve, it's really divided into 2 parts. There's an upper part that is the shoulder of the curve, and the lower part that looks like a straight line. It turns out that this part that looks like a shoulder is going to be the linear part of this curve, and the part that looks like a straight line is going to be the quadratic part of the curve. Now, that seems like it's a misnomer, but on a on a linear by linear plot, this looks linear, this looks quadratic. When we switch over to a semi-log plot, this becomes linear, this becomes quadratic. So we often refer to this as the linear part of the curve. We often refer to this as the quadratic part of the curve. But by mathematics, when we're looking at a quadratic curve in a semi-log plot, it does form a straight line. So this makes sense and fits in with our linear quadratic model. We are going to use these survival curves for lots of things. We're going to look at them to understand oxygen effects, survival in the cell cycle, repair, different kinds of repair that go on. So the survival curves are going to become important to us. So understanding them is also of big value. Okay, so let's start out looking at the linear quadratic model of the survival curve, which depended upon, if you remember, constants alpha and beta. Here is log of the surviving fraction against radiation dose, and I just want you to focus on this curve right here for a moment. This curve is the curve for X-rays. It would be very similar for gamma rays. As I said, two parts of the curve, one that looks like a shoulder, one that looks like a straight line. This is the linear, this is the quadratic. Now, what did we say about the mathematics of the linear part of the curve? That it relates to dose, whereas the quadratic part relates to dose squared. And what else we said at the time was that it was reliant upon these two constants that were alpha and beta. So the mathematics that were applied to the curve are that the surviving fraction here is equal to the exponential number e taken to the minus alpha d minus beta d squared, where alpha and beta are constants and d is the dose. What is most important for you to glean, and what I need to say is, you cannot look at this curve and do these calculations. In order to understand alpha beta, we are going to need to do much later when we talk about fractionated responses. That's how alpha beta are determined, is based on fractionated responses. But what this curve can show you is what's called the alpha beta ratio, which for us is going to become very important, because the alpha beta ratio is going to define how a tissue responds to radiation. Alpha beta ratio, this definition you got to know. Because it relates dose to dose squared, it is a dose. Always remember it's a dose. Sometimes on the boards they get it wrong by saying it is the it is the number at which, it is the fraction at which, it is the whatever, but it's a dose. So alpha beta is defined as a dose at which points the linear and quadratic contributions to cell killing are equal. It's where the alpha component meets the beta component on this survival curve. So here's the curve. Where do alpha and beta meet? Right here, we go down, we read the dose, it's 8 gray. So the alpha beta ratio for this particular cell tissue type or group of cells is alpha beta, is 8. There are a couple of other definitions of alpha beta ratios. It is frequent to be asking that question on the boards. This is the most common definition, but there are a few others that we will look at in a little bit. Okay, now I want to throw in what we see here for the neutrons or alpha particles. If you remember, I said to you earlier that there is low LET radiation and high LET radiation, that neutrons and gamma and alpha particles are considered high LET, high density radiation, very damaging per unit dose, versus gamma rays and X-rays, which are much less damaging per unit dose. And so we draw this curve, and what I want to tell you about this curve is there's no alpha beta ratio drawn here. Why? Because for high LET, all of the killing or the majority of the killing, 99+% of the killing, is by the alpha mechanism, not by beta. So this is going to be important to us. I'll talk about that in a moment, but I just wanted to note that nobody ever talks about alpha beta ratios for neutrons or alpha particles, because for densely ionizing radiations, the alpha is so very, very high. The beta is not 0, but it's approaching 0. So the alpha beta ratio is outrageously high. You can't even see it on this curve. Whereas for low LET radiations, you can actually see a distinct alpha beta ratio here. Okay, let me just see if there's, yeah, okay. So now so this is the first model, the linear quadratic model. There is a second older model that is not used that much, but one value in it will be particularly used, and it's called the target theory model. The target theory model was developed in the 1950s. It was developed by Puck and Marcus, and you've got to know their names. This paper is one of the most quoted papers in the literature. They too created a survival curve logarithmic fraction against radiation dose. These happen to be for HeLa cells, which are a cancer cell line, and they found a portion of the survival curve that had a shoulder and a portion that looked like a straight line. Let's look at the straight line part first, because it's maybe the easier for us. A straight line is defined by any 2 points. So in this example, they chose a dose that caused 37%, reduced survival by 37%. Why 37%? Because 0.37, 37% is the logarithmic number e taken to the -1 is 0.37. The natural log of 0.37 is equal to -1. So what they said was, this is about the dose. The D0 dose is about the dose to hit each cell in the culture one time, to cause one track of damage in the cell, in the culture. So how do you read the D0? You go to, in this curve, 0.01, 0.0037, you read the dose in between. It ends up being about, 110 or something like that here, 110 centigrade. And so this now is the D0. Would that D0 be the same if you looked at it up here from 0.1 to 0.037? The answer is yes, because this is a straight line. If you pick those portions of the curve, you would get the same result for D0. What about if you took it from up here? Would it be the same? The answer is no, because this is not a straight line portion of the curve. This is the shoulder portion of the curve. So it would look very different. So as long as you are on the straight line portion of the curve, if you go from 0.1 to 0.037, 0.01 to 0.0037, out here 0.001 to 0.00037, it will give you the same dose in between, which is, in this example, approximately 110. Okay, so that is the D0 dose. That is what's used. People frequently talk about it. The second part of the target theory model, I'm not sure a lot of people consider it valid. It is defined by 3 values. Value number 1 is the D1, which is the amount to reduce survival by 37% in the upper part of the curve, 1 to 0.37, and you read the dose in between. And it certainly is different. It's actually a little bit under 100 here. The second is what we call the DQ, the quasi-threshold dose, where you draw an extension line out, and where this intersects the x-axis up here, this gives you the DQ. Where this intersects the y-axis up here gives you the N. So let me say a few things about those. I honestly have not seen boards questions that ask about DQ or D1. I have seen boards questions that ask about N and D0. Why? How? D0, because it defines how sensitive a cell line is. If the D0 is high, the curve comes out like this. If the D0 is low, it comes in like this. N, which is where you take a line and extend this curve out. And if the shoulder is big, N is big. If the shoulder is small, N is small. So they often use it to talk about how big a shoulder the survival curve has. Okay, let's look at a few examples. Yeah, let me just say this. You'll remember last time we talked about the kinetics and how there are these direct and indirect effects that are happening within the first nanoseconds. I just want to point out that these survival curves that we're monitoring are taking place weeks after that initial exposure. They're the consequence of it, but they're still taking place weeks after exposure. Okay, so what's a survival curve? It is defined as the quantitative relationship between dose and the proportion of cells that divide. Puck and Marcus developed it. Our measure of cell death is the loss of reproductive integrity. The target for cell killing is the nucleus, its DNA. And now we come to this target theory model. Single hit, single target, single hit, multi-target. I don't think you need to know anything about the target theory model except what does a single hit, single target curve look like, and what does a single hit multi-target curve look like, and when do you find them? So here we go. Here's the single target, single hit curve. Here is the single hit multi-target curve. When did we just see each of these? Oops, sorry, I skipped it. Here it is. This is single hit multi-target curve for low LET radiation. This is single hit, single target. So when people talk about single target kinetics or single hit kinetics, this is the curve they're talking about. There's no shoulder on this curve. There are only 3 times when you see a curve like this. We'll talk about each one of them when they come up, but let me tell them to you now. They're on the boards. 3 times when you have single hit kinetics in cells. Number 1, if you have high LET radiation. Number 2, if the cells are in M phase. Number 3, if the cells have a defect in their DNA repair. That's when you see a straight line like this. So that's the single hit, single target kinetics. Here are some examples: single hit, single target, multi-target, linear quadratic model, 2-component. The linear quadratic looks most more like the multi-target, but the single hit, single target, this is what this looks like. This is what they're referring to when they have single hit kinetics, and it is exclusively in those 3 situations I just described. Now, here's the target theory model. Here's the curve for it. The log base E times N is equal to dq over d0. So we have these 3 values, N, dq, that are defining the upper part of the curve, and d0 defining the lower part of the curve. These are often used, but I've never seen it on the boards. But I have seen this next part on the boards. So what are the assumptions of target theory? What assumptions did they make? Did Puck and Marcus make at the time? They said that 1, there are ionizations that are taking place in your molecules that cause some sort of structural change. Turns out we know now it was DNA damage. Molecules were DNA and it was causing DNA damage. They said number 2, that there is a mathematical relationship between dose and effect. And the third thing they said is that each ionization event occurs randomly. Therefore, if it is random, it follows Poisson distribution, not a normal distribution or many other kinds of curves. The single hit, single target is linear when plotted on semi-log. Cells in M phase will be close to having that model damaged by high LET radiation. And I added in today that damage in cells that are missing DNA repair would also fit that. That was on the boards for the first time year before last. Obviously, there was nothing last year, so the last boards that existed. So you could actually do this formula for single hit, single target kinetics where survival is equal to e to the minus d over d0, relating number of cells surviving a dose, initial number of cells, and the d0 dose of the survival curve. For single hit, for multi-target single hit activation, it would include this formula, which is 1 minus e to the minus d over d0, all taken to the nth power. Now, we will look at some examples that take these into consideration as we progress through this, so that will maybe make more sense. I have some information here on target theory. I don't really think you need to spend a lot of time on it, but one thing, there are several things to know, and that is that high LET radiation produces these linear-like survival curves, that when we're using these curves, the D0 is the survival is going to be at 0.37. That's the definition. That these are based in a Poisson distribution possibility. I don't think you need to know all this about all this work, but let me explain Poisson statistics for just a minute, because it's also going to be related to us when we talk about probability of tumor cure. So we know that one lethal event is enough to cause the cell to have reproductive killing, where it cannot reproduce anymore. But the average of one lethal event per cell is really a pretty small number. So we use Poisson statistics to predict what number of lethal events there are per cell. If you had a normal distribution, this is what this would look like. This is what you're used to seeing, this sort of normal distribution that we use for grading, and we use it in this example for blood pressure. But the Poisson distribution is a skewed distribution curve that tends to look a little bit like a line on the far side, but has an abrupt peak at the beginning. So the old raindrop analogy is this. If you had 100 squares and 100 raindrops, if all the raindrops fall uniformly, you get 100 squares wet. But if the number of raindrops equal number of squares, 63% of the squares will be wet and 37% of the squares will be dry if it occurs randomly. So the D37 or the D0, the dose at which we get 37% killing, is the dose of radiation to reduce cell survival to 37% of the original cell number. This is more information about these curves and shoulders. I don't think you really need to know all this. This points out what I've already told you, which is the D0 dose, the N extrapolation number. But I want to point out one particular thing about this survival curve. Log of the surviving fraction against dose. This is high LET and low LET. Low LET has an extrapolation number. It has a quasi-threshold dose. It has a D0. High LET radiation has a D0. But look at this, the N is 1. The quasi-threshold dose is not detectable in this example. So curves with a shoulder have all those features, but curves without a shoulder do not have all those features. So wait, here we go. This is the curve I want next. So let me now compare and contrast these two models. The linear quadratic on the left, which if you're asked, the linear quadratic is the most accurate. The target theory, which is the older model on the right, but which there are some elements that we currently use. The curves are identical. It's how we think about them that is different. In the linear quadratic model, everything relates to alpha beta ratios. This model applies much more for irradiating tissues, cell mixtures, many different cell types in the body. On the right-hand side, the target theory model was developed in one cell type, the HeLa cells, and it applies much better for single cell types that are being irradiated than those that are being irradiated in the body. So what we're going to tend to see is, when we get into tissue effects later, we're going to tend to see a focus on alpha beta ratios. But We're going to focus on D0 and target theory model much more when we talk about cells and culture. What are the kinds of questions you might see on the boards that relate to this? Two cell lines have two different D0 values. One has a D0 of 1, one has a D0 of 2, which are more radiosensitive. Of course, the lower the D0, the more radiosensitive. Two qualities of radiation go to irradiate a single cell type. One causes a D0 of 1, one causes a D0 of 2, which is more likely to be high LAT radiation. And of course, it's the one that gives the lower D0 dose. There's another question about N. There are two qualities of radiation that are giving, there are multiple qualities of radiation that are giving values of N that are 2, 1, 0.5, 0.8, and 1. Which one is most likely to be high LAT? The answer is 1. So those parts of the target theory model you need to know. I haven't seen a lot of calculations with it, but you do need to understand these parts of the curve and these comparisons. We'll do a lot more of this as this course progresses, and it will become familiar to you. One of the reasons why the cell survival curve is related to the linear quadratic model is because if we look at the number of lethal aberrations, which in this example is actually dicentrics, and relate it to along the surviving curve, the answer is, look at the correlation here, it's virtually 100%. A few things that are a little bit off, but not much. With this kind of correlation, this would suggest that the dicentric model that we had developed in our last topic could now be applied to the survival curves that we see here. And this is why the linear quadratic model became useful for us clinically. And just to refresh your memory, this was only like a half hour ago, but it looks like this. If you remember the low dose part, radiation comes through, causes 2 track injuries. This is the linear. This is the quadratic. For high LAT radiation, because it's so penetrating, because it's so damaging per unit dose, most of the damage is using this method, this linear method. Very little requires multiple hits in order to generate it. Unlike low LAT radiation, where the majority of the damage is actually from the beta component here, the multiple hits killing, versus the alpha component, where it's a much smaller, in most examples, much smaller percentage. And in fact, many people would argue that when we're trying to kill a tumor cell, what really matters is that the 2 gray fraction that we're using must be in the quadratic part of the curve for that particular tumor. And it is in almost every example. We'll look at that in a little bit. Here's just some more information about the linear quadratic model. I don't think you need to know all this, but let me now relate all this to dose response curves. So this is a plot of the number of dicentrics per cell against dose, looking at multiple different qualities of radiation. These are gamma rays here, X-rays. These are neutrons, which are high LAT, coming from a californium source. These are americium, alpha particles. And you can see as the LAT goes up, the number of dicentrics per cell becomes very linear, not very curvy, and causes a great deal more cell killing per unit dose. And that is, in a way, the definition of high LAT radiation. And as I said, we have a whole section devoted to that. There will be a dose rate effect that we're going to see, where high dose rate gives us many more dicentrics per cell than low dose rate for gamma rays. For neutrons, that's more questionable. The dose rate effect is more questionable for high LAT radiations. So just to kind of review this, in the linear quadratic model, there are 2 components to dose. 2 components, one proportional to dose, one proportional to dose squared. Proportional to dose relies upon the constant alpha. The the portion proportional to dose squared relies on the constant beta, and where the dose of the killing from the alpha component and the beta component meets or is equal is considered to be the alpha beta ratio, making cell killing be an exponential function of dose. This kind of says the same thing, but now I want to talk about another definition of the alpha beta ratio, which probably won't make much sense now, but will make more sense after we talk about repair. But let me say it now, and then I'll say it again when we come to repair, and hopefully it will make sense. So this is the same formula, except that it's written slightly differently. It's the natural log of minus the natural log of the surrounding fraction. It would be e to the minus alpha d plus beta d squared if it were just the surrounding fraction here. So what is another definition? We already said it's the dose in gray, and I should mention always that it's a dose in gray. It is not in centigrade, it is not in rad, it is in gray. So that's an important part of the alpha beta ratio. It's the dose in gray at which the contribution to killing from alpha is equal to beta. Some people say that alpha is non-repairable damage and beta is repairable damage. Let's look at why that would be. Let's look at the image. Here we go. Why is alpha not repairable? Well, because you have a single track coming in causing these two damages. There's no time between the two of these in order to repair it. So these guys now can will be non-repairable lethal events. In the quadratic portion of the curve, theoretically, these two events are separable. And if it would be that you gave the radiation at low doses, at low dose rates and fractionated it, you could give this, wait a little while, give this, and allow for repair to take place. So this is repairable damage. This is not, because these two events cannot be separated, whereas these two events can be separated. I hope that makes sense. If not, please bug me and I can explain it again, but we will look at it again later in another section. So if it doesn't all make sense here, hopefully it will. So coming back to what we talk about then, alpha beta can also be defined as a dose in gray at which the contribution from non-repairable damage is equal to the contribution from repairable damage. And alpha is the single track component where the cell is inactivated after one electron track causes two breaks. These two events are not separable, they're one event, whereas beta is where there are multiple electron tracks, and they are therefore separable in time, and so therefore it's possible for repair to intervene in that second set. We don't need to talk about this. This is again the same formula with the same definitions, just so you have it. And here is the survival curve relating to whether it's high or low LET, relating to relative biologic effectiveness. The point I want to make here is log of the surrounding fraction against dose. The curves we've seen for neutrons have been straight. This curve has a little bit of a shoulder, and I do want to point out that some neutrons do have a little bit of a shoulder. So don't assume that it's totally shoulderless unless you're told so, because there can be a shoulder for some neutron survival curves. This is the original data from Huck and Marcus, and you can see how this curve looks. Now, I want to mention one other trick question I've seen on the boards before I go into the fractionation response, and it's going to relate to two survival curves that we already looked at. Here we go. Okay, so you're given the survival curve, and you have to calculate the D0. And they draw on this three lines. One from 1 to 0.37 here, one from 0.1 to 0.037 here, and one from 0.01 to 0.0037 here. And they say, they call this A, B, and C. And they say which ones of these represent the D0 in the curve? Is it A? It's not A. This is the D1. This is on the shoulder part of the curve. It is not a D0 dose. But B would be correct and C would be correct. So you would answer B and C for this part of the curve. Not A alone, not A, B, and C, not B alone, not C alone. But if you had a different curve that looks like this, and they had A, B, and C, what would your answer be? The answer would not be A, it would not be B, it would be C only. Why? Because the D0 must be on the quadratic part of the curve. It must be on the part that looks like a straight line, and here it would be partially on the shoulder and therefore would not be the correct answer. So Hall's definition is actually not correct on his in the textbook. He says it's the amount that reduces survival from 0.1 to 0.037 on the survival curve. It works in some curves. It doesn't work in all curves. What you have to say is that it's the reduction by 37% in a part of the curve that is quadratic, that forms a straight line. I hope that makes sense. If not, please ask and I'd be happy to explain it further. Okay, so let me go to where I was. No, next. Okay, so everything we've talked about is for a single survival curve where the cells are given one irradiation. This is log the surviving fraction against dose. They're given one dose of radiation. Here's the shoulder, here it comes down. Here's the D0, the amount to reduce this curve by 37%. This is on the quadratic portion. So we can go from 0.1 to 0.037. Here we get it. This is a multi-fractionated regimen. And I want you to notice that every time you give a new fraction, you repeat the shoulder on the curve. And that effectively pulls out the survival curve, so the D0 becomes larger. So we're getting less killing if we give the same dose in a fractionated formula than if we give it in a single crack. So just look at this here. 8 gray, look at what we get with single dose, look at what we get with a multi-fractionated regimen. So you can say, well, why the heck then do we ever fractionate radiation? And the answer is, it gives us a lot less killing of the tumor cell. That's not our good news, but it gives us less damage to our normal cells. It allows our normal tissue to repair. And so therefore, we fractionate in order to spare our normal tissue. We'll talk about that when we talk about repair in our next topic. Okay, but there's another thing that's important about this curve, and that is this formula right here. Oftentimes, we need to calculate what it takes to give us a log of killing, or as Hall would say, a decade of killing. It's D10. It's the dose to give us a killing by 1 log in our population. You calculate the dose to give 1 log of killing by taking the natural log of 10, which is 2.3, and multiply it by the D0. So the natural, so in this example, this is what, maybe 2 gray. You multiply that by 2.3, and you get the amount to give you 1 log of killing. You would do the same thing on a multi-fractionated regimen as well. So it's to give you a log of killing, and therefore spare 10% of the cells. So this is what we're going to use in our later calculations when we talk about how many logs of killing it takes to get to kill up 10 to the 9 cells, for instance. Here is what this looks like. This repairable part of the curve gets repeated, and that's what gives us sparing when we give multiple doses. So this is what's called the split dose experiment. This is a split dose where the first dose is X-rays and the second dose is neutrons. I don't know that you need to know this in any detail at all. And this is the same thing shown again. This is the opposite, where it's high LIT first followed by low LIT. I've never seen a board's question talk about those split dose. The only ones I've seen are X-ray to X-ray, which is what we do clinically, or gamma ray to gamma ray, for instance. Okay, there was a board's question a couple of years ago that asked about the D0 doses of a variety of different cell types. log the strength fraction against dose. It compared up here, Deinococcus radiodurans, yeast, E. coli, and human, and asked you to put them in correct order. And these are the diploid yeast, not the haploid yeast. So the answer was the most radioresistant organisms on Earth that we know of are Deinococcus radiodurans. Humans are pretty radiosensitive. Certainly some of the other certain plants and stuff are more radiosensitive than we are. But this was the correct order of the ones that were on the boards. And just to put it in perspective, here's Deinococcus radiodurans, 18 kilogray out here, barely touching it. These are lived in thermal vents, apparently were exposed to high doses of radiation, and really, really changed the way we think about it. There's a lot of work that's going into what makes these guys be so radioresistant. Can we somehow apply it to humans and increase our radioresistance? It looks as though the repair mechanisms are not so different. What happens is the DNA is broken and can reassemble in a variety of different ways, and that's what makes it be so resilient. Okay, so oftentimes the D0 is called the D37. In Britain, they would call it the D0. But the question becomes, how much difference is there in D0 values from different species and different cell types within the body? And among mammalian species, the range is not so great. It goes from 80 up to maybe 200 for the most resistant cells. The most radiosensitive are a few radiosensitive cell lines, but this is not, it's a log's worth of difference maybe. It's not much more than that. Ends vary as well, but it depends on the cell type that one is comparing. So where do we find our highest D0 doses, our most resistant cells? Mostly from patients that failed treatment. So squamous cell carcinoma from a treatment failure has the highest D0 dose and is considered very, very radioresistant. Among the more sensitive, here are the normal cells, but if we go to patients that have the radiation sensitivity syndrome, ataxia telangiectasia, that we'll talk about later in the DNA repair section, they go down to 0.5 centigrade. That's probably the most sensitive mammalian cells that we know. So, the range is maybe 2 up here to 0.5 down here, not a huge range, as we'll here say for chemosensitivity and chemoresistance, which has a very big difference. And here are just some of the radiation sensitivity disorders. Ataxia telangiectasia is going to be one of the most important, pneumatic breakage syndrome, Fanconi's anemia, etc., etc. Okay, so now I want to move from cell survival curves to talking about different mechanisms of cell death. There are a lot of boards questions that focus on these different mechanisms. This table is, there are a few others I would add to this table today, but it's all accurate and worth learning. What does it point out? The type of cell death, what things you would see changing in the nucleus, what you would see changing in the membrane, what you would see changing in the cytoplasm. Is there a biochemistry that marks it as a feature, and how do we detect it? So let me point out the highlights. Got to know for apoptosis that there's chromatin condensation that takes place, nuclear fragmentation, and DNA laddering. We'll talk about those in a moment. We'll also talk about the membrane blebbing that occurs, the formation of these apoptotic bodies where the membrane, where the cytoplasm just fragments off. That is a caspase dependent event, that it uses tunnel staining and annexin staining, caspase activity to be able to monitor it. Autophagy, mostly there is some blebbing that takes place. It forms these autophagocytic vesicles inside, and these go up, and they are caspase independent mechanism. Necrosis is just really an unscheduled cell death. There is a random degradation of DNA. The cell just swells and breaks open. There are no real chemical tests for it, but using electron microscopy, you might see cells kind of broken open. A feature of necrosis is going to be that because all the contents of the cell spill out, you often can detect inflammation there. So that's important. Senescence is going to be where the cells reach a point where they just can't divide anymore, but they're still alive. They're just not capable of division. Usually there's a flattening out of the cell's surface. There is an SA beta gal activity that becomes activated and becomes a marker of senescent cells. For mitotic catastrophe, this is a frequent cause of cell death. We'll look at how it interfaces with apoptosis and necrosis in just a moment. In a way, mitotic catastrophe is not so much a mechanism of cell death as a timing of cell death. Oftentimes, the nucleus fragments, dicentric chromosomes can be found, and it is caspase independent, and we can see tunnel assays sometimes to be positive with this. So here's how mitotic catastrophe comes in, and I think most radiation people would say that the majority of cells following radiation exposure die of mitotic catastrophe. What does it mean? Well, the cell gets damaged, and in the process of attempting its next mitosis, it can't do it all. So it dies, and this death can be from apoptosis, senescence, autophagy, or necrosis. It could also die prior to mitosis using apoptosis, senescence, autophagy, or necrosis. This is less common. This mitotic catastrophe is much more common. And this is it relates to cell survival curves because the cell metabolizes, it goes to divide, and it can't. So it can't form a colony, but it is still metabolizing. So therefore, in our assays of clonogenic survival or surviving fractions, we would not see these as living cells, even though they're still carrying out a lot of cellular processes. And here's just an example of one cell that was tracked through a different number of different processes where it went through apoptosis. Wait, let me just say, and this is another group of cells that were also tracked and were shown to follow many different kinds of mechanisms of cell death. Let me relate this to dicentrics because we talked about that too, is that after irradiation, dicentrics go down in number. Why? Because those cells are dying, and they're probably dying of mitotic cell death or mitotic catastrophe. The ones that had reciprocal translocations did not lose that much DNA. They are remaining large number because they did not undergo mitotic catastrophe. So if one thinks about like the dicentrics, the rings, and the interphase bridge, they're lethal aberrations, and that's mostly because the cells die in the process of undergoing mitosis or mitotic catastrophe. Necrosis is a common form of cell death. Mitotic catastrophe would be next, and mitotic catastrophe can lead to all these other forms of cell death as well. So I think it's kind of wrong to classify mitotic catastrophe in these all, although it does in many different textbooks. This is from a pathology textbook. Now, there are a few I haven't talked about that I'm worried are going to be on the boards, but I haven't seen them yet. One is called necroptosis. This is a programmed form of necrosis. So it kind of is, I don't know, I don't think it's a good naming because necrosis is supposed to be an unscheduled cell death, but they say it's like necrosis because it is inflammatory, but it is programmed, and so therefore like apoptosis, but its program does not involve caspases, so it's caspase independent. It is most often part of a viral defense mechanism to make cells undergo some form of suicide when the cell's infected. But it has been found following radiation exposure as well. It's poorly understood. Here is the process with TNF receptor and this rest in peace kinase, the RIP kinase, which seems to be the most important feature of necroptosis signaling. There is another mechanism called pyroptosis, and it's an inflammatory form of apoptosis, mostly associated with with a fever response, hence the name, so that you get infected with a microbial infection and you turn on inflammation and you undergo apoptosis. The immune cells release this giant cytokine bolus. It attracts other lymphocytes to come fight the invasion. Now, one of the things that's interesting is that flagellin has been shown to induce pyroptosis. Flagellin is actually a radioprotector in some examples. So, there seems to be a tie-in with radiation effects. It involves the release of these so-called DAMP proteins, the damage-associated molecular pattern molecules. So this appears to be important. And here is the pyroptosis pathway that involves NF-kappa B, which is also associated with radiation induction, inflammatory cytokine induction, which are also found with radiation. So there's some tie-ins to this that's not clearly known. Here is a summary of apoptosis, pyroptosis, and necrosis, comparing them all. Again, I haven't seen pyroptosis on the boards, so I'm just saying. Then there's a third kind called pheroptosis that is becoming very important in the cancer literature. I suspect it's going to make its way into the radiation literature very soon. There's a programmed cell death that is dependent upon the presence of iron. It occurs in tumors, in kidney injury, blood disease, etc. It does not have features of necrosis or apoptosis. What happens here is there's shrinking of mitochondria. The cristae of the mitochondria vanish, but there's a reactive oxygen species component, which for us is going to be very much related to radiation. Here is one of the mechanisms of pheroptosis, which involves a variety of proteins. And you'll notice that there's thought that PUMA is involved here, which is important in apoptosis, and this heavy iron dependence and that leads to the end. And here is a comparison of pheroptosis, apoptosis, autophagy, and necroptosis side by side. Again, I have to tell you, I haven't seen any of those on the board, any of those latter three on the boards yet. Not necroptosis, not pheroptosis, not pyroptosis, but I'm afraid it's going to be there, and that's why I'm trying to prepare you for what will be important. Okay, let's look at the other ones, which are on the boards almost guaranteed. Autophagy became popular on the boards about 3 years ago. It's been known for a long time, but nobody in the radiation literature cared about it that much. This is a comparing and contrasting of autophagy with heterophagy. What is heterophagy? It's the typical phagocytosis. Something gets taken in by the cell digests it up, it chews it up in its lysosome, spits it out as an exocytosis, and that's how we handle stuff that comes in from the outside. But in autophagy, the cell decides to eat itself from the inside. So it is a form of suicide. The cell starts eating up, in this example, the Golgi apparatus, puts it into the lysosomal body, eats it up, and spits it out. So this is a process of doing it from the inside. And here's an example of where there is a mitochondrion sitting within a lysosomal body. So it's eating up its own mitochondria in this particular example. For apoptosis, there are tons of questions on the boards. There's just so much known. And the reason why a lot of people go with apoptosis as being important is because it's thought that we can modulate apoptosis and lead to even more cancer killing than we're doing now. So there is mitotic cell death, where the cells die to the next division, and apoptosis can be a part of that. It's a programmed cell death. It is a usual part of embryonic development. It does also occur in T cell differentiation. Without apoptosis, we don't get development of the normal embryo. It affects scattered cells here and there. I'll explain what that means in a minute. It uses energy. It is a form of suicide. So the cell actually has to make a commitment to have proteins synthesized in order to kill itself. There's no tissue disorganization that happens at all. The dead cells are just sort of squeezed out. Here's the membrane blebbing portion that I mentioned before. The cell is a lymphocyte that's undergoing apoptosis. It's blebbing out its cell contents. The cell contents are in these little blebs that are released, and they're called apoptotic bodies when they're released, and they're often phagocytosed by the neighboring cells to be used. Here's the mechanism. So here's the radiation causing the injury. In this example, it's causing DNA damage, but you can also have membrane activation of TNF and its receptor, which is also induced by radiation. These each induce different effects, but all lead to this execution caspases. The caspases then cause breakdown of the cytoskeleton. They cause endonuclease activity in the nucleus. DNA gets broken and fragmented. All of this buds out in a cytoplasmic bud, forms this apoptotic body, which is phagocytosed by the neighboring cells. Now, what is cool about this mechanism is that the cell contents are never spilled out. So there's no inflammation that comes about because of apoptosis. This is another mechanism, another pathway. We're going to have to learn some of these caspases later. We'll talk about them later. But you're going to have to know the caspase 9 activation of 3, 6, and 7, caspase 8 activation of 3, 6, and 7, the mitochondrial activation, the so-called intrinsic DNA damage pathway, the so-called extrinsic TNF, or fast ligand pathway that also operates here. But the key here is that there are caspases that are turned on, and the caspases initiate the breakdown of the cell, membrane blebbing, and the apoptotic body formation. This is again showing the same thing. Okay, so let's compare and contrast apoptosis, which is an active suicidal death, with necrosis. What do you have to know here? What you have to know is that apoptosis involves these blebs that now form apoptotic bodies that are phagocytosed. No inflammation. Here is the apoptotic cell all by itself. It's scattered. You don't find a cluster of apoptotic cells. There's one here, there's one there, there's one someplace else. Necrosis, on the other hand, causes the cell to swell up, throw out all of its material, spew it forth. This attracts lymphocytes, causes an inflammatory response. Lots of cells die at the same time. So this appears as a cluster of dead cells instead of scattered here and there. The second thing that we're going to talk about is that in apoptosis, the DNA is broken apart in a very orderly fashion, whereas in necrosis, because it's unplanned, the DNA is just degraded and falls apart. We'll talk about that. I'll show you an example of that. Okay, I think I said that already. Here's comparison. Necrosis, the cell swells. Apoptosis, it shrinks because it's sending out those blebs. The necrosis, the DNA becomes less intense. There's a general DNA degradation, whereas in apoptosis, it's laddered. It forms in a very orderly fashion, generally around nucleosomes, causing degradation into like 185 base pair units. In necrosis, there's increase in membrane permeability, whereas in apoptosis, there was this membrane blebbing. In necrosis, there's no need to have any new synthesis take place. In fact, the cell's trying to kill itself, the cell's dying, there's no time. Whereas in apoptosis, the cell's trying to kill itself, so it turns on an entire program of cell death, and is very active metabolically, including calcium pumps are still going, ion pumps continue to function in apoptosis, not so in necrosis. Now, I need to point out that a lot of the same things that cause apoptosis also cause necrosis. So radiation, it doesn't just cause one or the other, it causes both. Hypoxia doesn't cause just one or the other, it causes both. So it's not an either or thing for apoptosis and necrosis. Often the same things that induce one induce the other. In the apoptosis world, there are pro-survival proteins and pro-apoptotic proteins. You have to know that BAX, NOXA, PUMA, BID are pro-apoptotic. When you get lots of them, the cell gets geared toward apoptosis. BCL2, BCLXL are pro-survival, and when you get lots of them, the cells lean against apoptosis. The difference between these proteins is this one domain, the so-called BH4 domain, which is found in the pro-survival, but not in the pro-apoptotic cells. One of the main features, we looked at it, but I didn't really emphasize it, is going to be induction of P53, which this is no longer a dotted arrow. P53 turns on both Noxa and Puma, gives us this mechanism that leads to caspase activation. And this is pretty much the same thing, balance between them. Now, I don't know that there's a board's question that asks this, but I believe that knowing this helps you think through some of the board's questions and maybe even some clinical issues that might come up. So what's the importance of apoptosis in different kinds of tumors? In lymphomas, apoptosis is very, very important. More than 90% of the cell killing in apoptosis of lymphomas can be from apoptosis. In carcinomas, it's moderate. It's maybe 50-50 as to whether you're going to find apoptosis or not. But in sarcomas, it's virtually absent. And this is going to mean that, like, you know, a lot of times because cells are not dying in apoptotic death, they're not shrinking. So frequently you might not see that a sarcoma shrinks in response to radiation. With lymphoma, you will see it shrink. It doesn't mean the radiation is not working. It just means that probably apoptosis is not in place. I think I said all of this except the latter's story that I've alluded to, but I need to mention in detail now. I have a diagram that I think is going to explain it better. Here's kind of the process and the steps in order for both apoptosis and necrosis. And there's another feature of apoptosis that we haven't mentioned that is not found in necrosis either, and that is the release of cytochrome C from the mitochondria. So what happens is when apoptosis is going on, protons get pumped out, and so is cytochrome C. So if we see a lot of cytochrome C within the cell, it's usually an indicator of the fact that apoptosis is ongoing. What induces it? Lots of stuff. Chemotherapeutic drugs, ionizing radiation, hyperthermia that we'll talk about in here, photodynamic therapy, lots of different things induce apoptosis in different situations. And so it's not just radiation. Okay, so now we're going to talk about the laddering and how this relates to cell survival. Let me do this first, and then I'll go back. So here's the laddering. When apoptosis occurs, DNases in the nucleus are released. They're endonucleases. Endonucleases cut within DNA, exonucleases cut at the ends of DNA. Endonucleases are activated, and these cut the chromosomes up. But we have these nucleosomes with 185 bases around them that are protected by proteins. The nucleosome is a group of proteins that are sitting on the DNA, protecting it. So when apoptosis begins, the DNases cleave around these nucleosomes, but they might create clusters of 1, 2, 3. So an apoptotic cell, if you were to look at the DNA in an apoptotic cell, you would see a ladder that differs by 185 bases in its size. So down here is 1 times 185, 2 times 185, 3 times 185, 4 times 185. So this laddering is characteristic of apoptosis. It's not found in other forms of cell death, and it's because of this very methodical degradation of the DNA. In necrosis, however, the DNA just degrades. It's a total smear. It degrades randomly, and there it is. So let's go back now and look at this. Here's the ladder for a variety of different cell lines, and some you can't see it so well, some you can. But there was a group that did, Don Chapman at Fox Chase, he looked at log of the surviving fraction against radiation dose, looked at survival fractions with apoptosis as a dominant means of cell death versus an absent means of cell death in a variety of different cell lines. And you can see these survival curves where apoptosis was absent, the cells are a little bit more radioresistant. Now, please do not think that, whoa, if a cell's missing apoptosis, it's radioresistant, or it would show up on a survival curve. These were specifically selected because of their apoptosis as a cause of cell death. But we know that radiation causes necrosis. It causes a lot of other forms of cell death other than apoptosis. So it can't all just be related to apoptosis, and it's not. Another assay that's used, and this has been on the boards, is an annexin assay. What annexin does is it detects phosphatidylserine on the cell surfaces as they undergo apoptosis. This is red fluorescence against green fluorescence. The green fluorescence is picking up the phosphatidylserine. The red fluorescence is picking up the cells that are dead. So here we have the healthy population. Here are the cells that have the phosphatidylserine on it. These are the cells that are dying as well. And so we use annexin as a means of being able to measure these apoptotic cells, radiolabeled or fluorescently labeled annexin. And here are apoptosis in a variety of cell lines, just saying, look, genotype matters. Okay, here are cells that are missing P21 here and have a higher apoptotic fraction. P53 would make a difference here. So genotype is going to matter a lot about apoptosis if they're missing or have lots of copies of pro or anti-apoptotic cell lines. So intrinsic radioresistance, particularly in tumor cells, has been tracked with the following molecular changes. And I would learn these because I've seen boards questions on them. Overexpression of the RAF oncogene is often associated with radioresistance. Having a point mutation in NH or KRAS, one of the RAS families, is also affected. It usually affects the G2 delay more than anything else, and therefore they become radioresistant. BCL2 overexpression is also very commonly associated with radioresistance, but is an anti-apoptotic protein, right? So if you have lots of BCL2, the cells don't undergo apoptosis. HER2/neu has also been associated with overexpression and radioresistance. And here's just the RAS example where log the strength fraction against dose, and you can see the differences in radiation sensitivity depending on the kind of RAS. Here it is with RAF, the same idea. So this is just demonstrating what I listed in that table before. Now, this is a kind of growth delay where we will talk about these kinds of assays later, where we measure the ability of a tumor to respond to radiation using radiation to see how much it delays the growth of the tumor cell. The only point I want to make here is that P53 makes a big difference in how the tumor responds to radiation as well. Now, there's another feature I want to relate to cell survival curves, and that is the tumor control probability. We will talk about this way later in this course, but I want to give you a framework for thinking about it in terms of cell survival. So one thing you should know, this is assumed on the boards that you know, is that a 1 gram tumor has 10 to the 9th cells and has undergone 30 doublings. If you get up to have a 1 kilogram tumor, probably the person's not going to be able to sustain that tumor in their life and they're going to die. So, 1 gram, a little over a gram, those are going to be what the body can sustain. We said this. If you have a little over a gram, 10 to the 10th cells, and you get 90% cell killing, you're going to be left with 10 to the 9th cells. You're going to be left with a 1 gram tumor. What about if you get 99% killing? You're still going to be left with a 10 to the 8th cell tumor. It's pretty hefty. Where we need to start thinking about for cures is down around 1 cell per tumor. That's about the kind of place where we need to think. So tumor cure probability allows us to think about that. I'm not going to go through all the mathematics of this now, but it is based on Poisson statistics. Again, radiation works randomly. Poisson statistics is what we use. The probability of a tumor cure is equal to that logarithmic number E taken to the minus n, where n is the number of surviving clonogenic tumor cells. So by definition, because the natural log of -1 is 37, the tumor control dose for 37 leaves 1 cell per tumor, by definition. is this person, so this is just, so this is a frequent question and you need to know this. Tumor control dose for 90 leaves 10 to the minus 1 cells per tumor, or what is 10 to the minus 1 cells? How do you leave 10 to the minus 1 cells in a tumor? It means that in 10 patients, you left 1 cell. TCD 95 leaves 0.05 cells per tumor, or 5 cells in 100 patients. TCD 99 leaves 10 to the minus 2 cells, or 1 cell in 100 patients. So when we talk about depopulating of tumors, we are going to talk about depopulating from a tumor size of 10 to the 9th down to, say, 10 to the minus 1th. How many logs of killing is that? That's 10 logs of killing. We're going to ask the question, if the D0 is X, how many D0s do we need to have in order to decrease the cell killing from 10 to the 9th down to 10 to the minus 1? Well, we're going to need 10 times 2, it's 10.3, 2.3 per times the D0. We'll talk about those and we'll do some example problems of that, but that's how this is going to play a role. And actually, here are the problems. Let me do my best to go through these now. I mean, because I think it finishes up the phase. I'm not sure I'll get to the very end of this topic. There's one other thing after this that I might wait for next week to talk about. Okay, so here we go. Exactly the question I was talking about. You have 10 to the 9 cells in the tumor. You have a dose response curve with no shoulder. So that means it's a straight line, right? The D0 is 3 gray. What does the dose need to give a 90% tumor cure? Well, what does a 90% tumor cure mean here? It means 1 cell in 10 patients. So if you start with 10 to the 9 cells and you go down to the 10 to the minus 1 cells, that's 10 logs of killing, or 10 decades of killing. You use this formula, 1 decade of killing is equal to 2.3 times the D0. In this example, the D0 is 3 gray, so it's 2.3 times 3, which is 6.9. That's the amount to give you 1 log of killing. We need 10 logs of killing. We need 69 gray. So this is a typical kind of question, and there was no reason why they had to tell you what the dose was or anything else. They just told you that to make you guess. Let's go to the let's skip that. Oh, we'll go back to that one in a minute. Here's the same problem as 1. 10 to 9 cells, D0 is 3 gray. What do you need for 90% tumor cure? But this time, the cells went through 3 doublings during the course of treatment. How much more dose do you need to add in? Well, what Hall says is the correct answer, is that 3 cell doublings means you increase by 2 times 2 times 2, which is 8, which is about a decade more cell killing. So what you would do is add in 1 more decade of cell killing, and you would get 75.9 as your total, or you need to add a dose of 6.9 gray. And here's problem 3, which is very similar to this. Now you're doing the tumor with 10 to the 9 cells, and you're giving 40 gray. The D0 of the tumor is 2.2 gray. How many tumor cells are left after you give 40 gray? Well, you do this kind of the reverse of what you did before. You say 2.3 times 2.2 is the dose to give you, take you down by 1 log. That's 55 gray. 40 gray is how many decades of cell killing? Well, 40 is 5 grays. It's 8 logs of cell killing. So if you start with 10 to the 9th, you decrease it by 8 logs, you get 10 cells are left. Finally, this is based on that 37 number. 10 to the 7th cells are hit with single hit kinetics. That means following a straight line, there's no shoulder, so that 1 hit per cell is taking place. How many cells survive? Well, the D0 is the dose that gives 1 hit per cell. So this is going to reduce the number of survivors to 37%. So you start out with 10 to the 7th cells, you multiply that by 37%, you get 3.7 times 10 to the 6th. So that's going to be the amount that you reduce. Now let's go back and pick up that one question that's out of order. This uses a different formula. It looks at surviving fraction using that 1 minus 1 minus e to the minus d over d0 to the nth power. If the d0 is 150 centigrade and the n is 5, what fraction of cells will survive 6 gray? You really just plug this in. The dose here is 6 gray. You have to make it them be in the same units, 600 centigrade, 150 centigrade, or you could do 6 to 1.5. It doesn't matter, it'll come out the same. You do 1 minus e, and then you take this to the nth power, which is here, 5, and you get a number of 0.1. I have to tell you, I've never seen a question like this on the boards, but it's there if you want to look at it. These are all questions from Hall's textbook. These are just some extra things I put in here to maybe help you. I've said all of this before, but it gives a little bit more about the D37 and the dose response and the shoulder of the survival curve and things like that. Alpha beta ratios, predictive assays. Okay. So let me just see where I am. I want to, yeah, I'm not going to get through all this. Let me go through predictive assays and then we'll call it quits. There are a number of people that try to predict how a tumor is going to respond to therapy, and actually how normal cells are going to respond to therapy too. And we will have a whole section on predictive assays, but there is one assay that is considered the clonogenic assay. It's actually the gold standard if you can do it. But it's a very long, tedious assay, as you can see. It takes weeks to get the data. So what a lot of people have done is they've done a bunch of other assays. These are not so good. They don't work so well. But this SF2 does appear to work pretty well. What is SF2? You have to know the definition. It's looking at the surviving fraction at 2 gray. It is not the perfect solution, but it works. Here's why. Here are 20 different human cell lines that were tested. Here are their survival curves. Log the surviving fraction against dose. Look at the 2 gray point. The 2 gray point is a predictor for how all of these will work. So you don't want to, you might not have the time to do this long survival curve for each one of these, but you can do a surviving fraction at 2 gray, and it is a very, very good predictor for low LET radiations. It's not going to work for high LET. In fact, in high LET, people would argue that it's 0.68 gray. I think even that's much more questionable. But for low LET, the surviving fraction at 2 gray works really well. We'll talk about like normal tissue effects later. We'll come back to this. We'll talk about it later. But this is a pretty good assay, and you got to know it's the surviving fraction at 2 gray, which is used as a predictor for how radiation responses are. There are other assays. We will not talk about these today, but they're here for you to take a look at them for predicting radiation injury, radiation accident, radiation injury within tissues, and we'll come back to that later. Okay, so when we left off last time, I wanted to bring up a final topic that I just felt like I couldn't handle in a very short period of time. It won't take longer than 5 minutes, but we didn't have that much time last time. And that is, we've done a lot on survival curves. We talked about the DNA being the most essential target, but we didn't talk about the proofs of it. And there are several boards questions that ask, how do we know that DNA, nuclear DNA, is the most important target for cell killing? And there are 3, perhaps 4 experiments that have been done that address that specifically. Let me go through them. The first is the use of a polonium wire to irradiate the nucleus plus cytoplasm or the cytoplasm only in a cell. What this looks like, if I can get the slide up, there we go, is shown here. This is where there is a polonium wire, and I don't know if you remember, there was that spy who was poisoned with polonium years ago in the UK. He had ingested polonium. Well, actually, his name was Litvinenko. We'll see some pictures of him. In any case, polonium that was given to him, the alpha particle was an alpha particle emitter. And alpha particles, if you remember, do not travel very far. They're stopped by a simple piece of paper. So what was done in this experiment was a polonium wire was placed at a particular distance from a cell and emitted alpha particles that hit only the cytoplasm. And then they moved the polonium wire over. It hit the nucleus, so they're hitting both the cytoplasm and the nucleus. And they asked which one kills cells. And the answer was, if the nucleus was hit, the cells died very easily. These were done in Chinese hamster ovary cells. The second experiment was done with a tritium label. Sometimes people talk about a tritium and iodine label, which is the experiment that I've diagrammed here. But in fact, the very best experiment was a tritiethymidine label. The tritiethymidine goes to the nucleus and labels the DNA. And then a tritiated concanavalin A, or Con A. Con A is a lectin that binds to the mannose residues on the surface of cells. And they would compare these. And so what that looks like is here, where they label the nucleus with tritiethymidine, which is a beta emitter, or they used concanavalin A on the outside of the cell, and they asked which one gives better killing. And here are the data shown with the iodine, but the results are the same. If you get the nucleus, you get lots more killing than if you get only the cytoplasm. Last experiment is one that comes up frequently because it's an important tool in the field. It's the microbeam. There are only a few of these in the world right now. Columbia has, I think, one of the ones in the US. There's one down at Texas A&M, and then there's one in Belfast in UK. Anyway, the microbeam looks like this. And we're going to talk about this setup later in a series of discussion about low dose effects. But here we go. This microbeam puts a particle beam or an X-ray beam through a cell. And it does this by having a collimator that's so fine that you can hit either the nucleus plus the cytoplasm or the cytoplasm only. If both are hit, it takes only one hit to kill the cell. If only the cytoplasm hits, it takes between 49 and 67 hits to hit the cell. And one wonders whether it's just that the accuracy isn't that great, and that's why it takes multiple shots in order to kill them. So this was good evidence that nuclear DNA was the most important target. Finally, there is a study that looked at a correlation between DNA damage and cell killing, and those things that caused double-strand breaks give much better cell killing than those that don't. Examples here, some double-strand breaks were caused by hyperthermia. That was not because hyperthermia by itself causes double-strand breaks, but because it was inhibiting repair of double-strand breaks. High LET radiation does, and that was good evidence that if you can get double-strand breaks, you can get cell killing. And we will continue to talk about that throughout here. These are just the survival curves, just showing you again what they look like. And here's the summary from the entire section that we talked about last time. Cell survival curves are used to assess how sensitive a particular cell line or any cell type might be to radiation. They have established components that help us define the shape of the curve, and it's those components that we actually use as mathematical models. And finally, the survival curve data are consistent with nuclear DNA being the most important target of radiation damage to the cell. Lots of other things get hit, including mitochondrial DNA, but nuclear DNA is the most